In this video series, we're going to look at seedless vascular plants. So these are our kind of successors to the bryophytes, and because there's no longer this open, barren terrestrial surface for them to colonize, they have to compete with other plants. And that competition is going to be for access to sunlight now, so we need to grow tall. Here's some of our diversity of seedless vascular plants. Most of the seedless vascular plants that you will see on the day-to-day basis will be ferns and horsetails, but there are a diverse kind of assemblage of these plants. In this video, we'll look at the characteristics that unite the seedless vascular plants, and we'll look at some of the currently living groups. The first group that we'll look at are the club and spike mosses, the Lycopodiophytes. On the right, you can see a picture of a staghorn fern. These are epiphytic ferns. They grow on other plants and they get enormous. Sometimes they can get so big that they will break the branches of the trees that they grow on. If you ever go to the HSU greenhouse, you'll see some really cool examples of staghorn ferns. You can also find them at plant stores in the area. They are fun for people to kind of cultivate because they can get so huge. So let's make some additions to the big phylogeny. So when we left off, we had done all of the groups leading up to plants. So I'm just going to have plants on this particular one. So we have these 5 traits that lead us up to the group of plants that unite plants, our synapomorphies. You would find alternation of generations in this. You would find terrestrial, you'd find multicellular, all those ones we covered before. So I'm just going to dot those. So the first group of plants were the bryophytes, and I'm going to have these as an unrooted group. They are kind of unresolved. So we had the hornworts, we had the liverworts, and the mosses. So all of these are lineages of bryophytes, and we don't know which one came first. In the paper that I'm using for our class, we have the hornworts coming out first, and then it goes liverworts and then mosses. But that is not something that we know for sure. So something that unites this group, we would have gametophyte dominance, and we start to see the evolution of a cuticle and stomata, but it's not in all parts of those plants. So I'm going to put those here, even though we see this happening beforehand. Because in our liverworts, we just have those simple pores, and we only have stomata on the sporophytes of the mosses and the hornworts, so it's tricky to put it into our phylogeny before. So we have a cuticle and stomata in all of our future sporophytes. We're also going to move to sporophyte dominance. So our gametophyte is going to get smaller, and it's going to only be this sort of thalloid flap of tissue that is still going to have the sporophyte grow out of it, but the sporophyte is now going to be nutritionally independent. It's going to be larger, and it's going to be the longer lived part of the life cycle. So when you look out and you see plants, most of those are sporophytes. We're also going to have sporophyte branching. And so that sporophyte branching means that instead of how a moss would just have one little capsule at the end of that seta, if we branch that sporophyte, we can have many sites of meiosis. So we are increasing our ability to have genetic diversity and to produce propagules that will have more options for how to deal with this stressful environment. So more sites of meiosis. And then the next cool thing is lignified vascular tissue. So we now have our tracheids. And those tracheids are going to allow us to transport water from our lower tissues to our higher tissues. And we have now true roots and leaves because we have vascular tissue. Is it leaves or leaves? I never know. I should probably look that up at some point. And we'll have two major groups of SVPs. We'll have Lycophytes. Well, I guess these are both Lycophytes or Lycopodiophytes. So I'll just break these into two major genera. Lycopodium and Janella. And then here we'll have ferns and horsetails. See, these are monilophytes. So we have monilophytes and Lycopodiophytes. And the difference is that here we have megaphylls, so larger leaves. And then we'll save the rest of this for our future groups of plants. So these are all bryophytes and these are all SVPs. So here's our characteristics that unite SVPs. We just listed them on our phylogeny. So lignified vascular tissue having those tracheids, and we'll also have sieve cells. Sieve cells won't be lignified, but they'll be part of this kind of true phloem that we've now established. So that helps us grow tall because now we can grow away from the water surface, right? Because we can transport water up through our tissues up to the aerial parts. We don't have to be growing just in the water. And for our photosynthetic parts, we can have those raised above the surface and then transport our photosynthates down to our roots. So that allows us to have this more complex structure and to grow tall. We also have a sporophyte dominant life cycle. So the sporophyte is now nutritionally independent in the longer lived part of the life cycle. It's also larger. And we have sporophyte branching. And that starts out as dichotomous branching, which means it splits in two equal parts every time it branches. And that's really obvious in the Lycopodiophytes. Ooh, and here it says they're paraphyletic. That was how we had those two different groups that don't share just one common ancestor. So that makes them paraphyletic. This common ancestor is shared by these SVPs and these SVPs, but also all plants after that. So that makes them paraphyletic because they don't all branch from the same point. If they branch like this, they would have this common ancestor here that they all shared, that no other groups of plants did, and that would make them monophyletic. But we don't see that. So most of the SVPs that we'll look at have rhizomes. They have these underground stems that allow them to reproduce asexually. And we'll see that in each group of SVPs for our four major lineages. So let's look at our Lycopodiophyta. These are club and spike mosses. They're called that because they sort of look like mosses. We have some Janella growing in our little terrarium in the classroom, in the lab room. But Lycopodium, I've only seen, I think you can get it at plant stores, but I've seen it growing wild in the community forest. I don't know if it escaped there, but if you go to Alaska, you'll see Lycopodiophytes everywhere. But here we see them only kind of rarely. So I'm going to switch. It's going to kind of be weird because I'm drawing the leafy green parts in purple, but that's our sporophyte color, right? So we have these kind of spiky sporophytes producing little leaves. So we'll just say that's what I'm drawing all here so I don't have to spend forever drawing it. These are microphylls. So they have just a single vein of vascular tissue. So if we were to look at them, they would, have that little midrib, but that would be it. And then at the end of the sporophyte produces this cone-like structure. This is called a strobilus. So if we zoom in on that strobilus, it looks kind of like a cone, or it has these little leaves that are arranged kind of all around it, but we're just looking at a cross section, so they look they're in a little ladder. And each of those leaves will bear a sporangium, and you can actually see these. They're macroscopic. And depending on which group of Lycopodiophytes you're looking at, that might be just a regular sporangium, or we might call it a megasporangium or a microsporangium. If we are looking at Lycopodium, which is more what I've drawn here, that will just be a regular sporangium and it'll be homosporous. So meiosis will happen inside here to produce spores to grow into gametophytes, and all of those spores will be the same size, and they will all grow into bisexual gametophytes that make both male and female parts. So they'll make antheridia and archegonia. So this one is homosporous. If I was drawing the Janella strobilus, we would see some sporangia with large spores. Those would be the megasporangia, making megaspores, and those grow into the megagametophyte, which would make archegonia. Or they could grow into the microgametophyte if they're small spores inside a microsporangium, and those would produce antheridia. So we have meiosis happening within the sporangia to produce homospores that are going to grow into bisexual gametophytes. And this will probably be this creeping stem that grows underground, overground. They tend to be kind of dangly and creepy, not creepy like creepy, but creeping. And so it'll produce another one above ground with a strobilus, but this will have roots growing out of it at different nodes. Because it's a stem, these roots are adventitious roots. From the rhizome. And that rhizome would be underground, producing these above ground shoots. The gametophytes start to get pretty interesting. So gametophytes in our mosses were the larger part of the life cycle. Here they get increasingly reduced. So gametophytes will either be photosynthetic and very small, or they'll be non-photosynthetic and parasitize other organisms. So that is what we have on the left, is a parasitic gametophyte from one of our Lycopodiophytes. Here's a cross-section, or not a cross-section, a long section through our Lycopodium strobilus. You can see that in each of these sporangia, the spores are, they're still developing, but they are not different sizes. This structure here is called a sporophyll because it's a leaf, a fill, that makes sporangia, so it's a sporophyll. And this is what it looks like macroscopically. So see how it looks kind of like a moss, where we have these spirally arranged leaves and it looks bushy, but then you get this strobilus on the top, so you can tell it's not a moss because of that. So in bryophytes, we only had one sporangium per sporophyte. Now we can have many. Within that strobilus, you have many sporangia, and then it's not that each individual only has one strobilus at the top. You can have an individual with many strobili, and each of those have multiple sporangia. So that exponentially increases your chances for genetic variation, your opportunities. If we look at Janella, so this is the one that we have in our lab room. Janella has microphylls just like Lycopodium. See these little tiny leaves that would have just one vein of vascular tissue. But inside the strobilus, we have megasporangia that produce megaspores on a megasporophyll. And we have microsporangia that produce microspores on a microsporophyll. So this is the condition of being heterosporous. After SVPs, everything becomes heterosporous, but we have homospory in most of our groups of SVPs. So Janella is sort of the only one to be heterosporous, but then that is convergent evolution with the rest of plants, because Janella is kind of a more ancestral lineage. Selaginella is another one that can dry out completely. It's one of these Lazarus plants that can dry out, sit, completely metabolically inactive, and then you rehydrate it years later and suddenly it's alive again. Very cool.